This conference will now be recorded.
Today's presentation is hosted by Heart Nursery, one of California's oldest and most respected cannabis nurseries. We're here to discuss their new patent-pending process for testing and eliminating cannabis dudding. A few quick reminders: if everyone could stay on mute and ask questions by messaging me directly using the chat function on your control panel. We also have a press release and some high-res graphics to go with today's presentation, so please email me at laura@lbqstrategies.com to receive any materials or for additional interviews after the call. And on the call, we have Dan Grace, the founder and president of Heart Nursery. We've also got Dr. Jeremy Warren, who's the director of plant health at Heart, as well as Will Roberts, who's the research manager at Heart. And Dan, would you like to take it from here?
All right, thanks a lot, Laura. And thanks everyone for tuning in. I'm really excited and super proud today to present this webinar with everyone. Our presentation is the culmination of almost 5 years of work now within the company. And I think it represents really the most important breakthrough improvement in cannabis crop health in the history of the industry, at least as long as I've been working in it. And it's always been a part of Heart's corporate DNA to tackle really big problems, not just for our company, but for the industry as a whole. That's why we've advocated so aggressively to legalize and regulate cannabis at all levels of government, why we've done big charitable works, and really nowhere is that sort of focus more keen than our focus on improving results for our farmer customers. Heart's mission is to help farmers thrive by making the healthiest, most productive cannabis plants available at a price farmers can afford. And we do that through disciplined commitment to research and continuous improvement. This is really our pinnacle achievement on that front. We first began looking into this issue almost 5 years ago. Growers had started reporting online symptoms that they called dudding, which were characterized by like stunted growth and big crop losses, big crop yield losses. They would get these plants that really didn't produce very much usable bud whatsoever, and there were very few cannabinoids or terpenes. It could be quite severe, resulting in very large partial or full crop losses. The symptoms later became described as putative cannabis infectious agent, or PCIA, which is what we've called it until now. And now that we've developed this test, we can tell with confidence that that this pathogen is very widespread throughout the industry, and it costs untold millions of dollars every year. And fortunately, though, we've also developed a cure to this disease. So without further ado, I'm going to turn it over to the scientists in the room and have them tell you all what we've found. So I'd like to turn it over first to Director of Plant Health, Dr. Jeremy Warren, who will tell you a little bit about his work to identify this pathogen, and then we'll hear from Will Roberts, who will tell us about how we've gone about solving it. So Jeremy, take it away.
Thank you, Dan. Yeah, I'm very excited to share this research and work we've done together over the past couple years, and hopefully help the cannabis industry out with this problem. Next slide. So as Dan was mentioning, this is a problem throughout the industry, and there was a survey done by, I believe, Dr. Crum, who went out to a few grow houses, outdoor grows, nurseries, and just took a survey. How many plants here are infected? How many look healthy? And none of them had zero problem with this disease, and some companies are up to like 33% infection rates. So that could provide huge losses for these companies. Next slide. So these are some common symptoms we see of PCIA. Nice healthy plant on the left. On the right side, there's a symptomatic plant. As Dan was mentioning, often these have a lot of lateral stemming on the bottom of the plant, yellowing of leaves, malformed leaves, and brittle stems. Next slide. And so here's a close-up of some of the foliar symptoms. On the far right, we consider these the classic PCIA symptoms. It's kind of yellowing of leaves, the malformation of leaves. There's also some things we see in the greenhouse, this middle slide, this bright yellow sort of symptoms. We consider that to be genetic in nature. So we wanted to test, so we're going to look at what might be causing this one as well. So next slide. So how do we do that? How do we find out what sort of pathogen could be causing the disease? So there's a really cool technology out there called RNA sequencing. And what this allows us to do is get a snapshot of the microbiome of the cannabis plant. It'll tell us all the organisms that are on the outside or inside of this plant and help us figure out which one of these may be causing the disease. The way we do that, take a cannabis leaf, grind it up, extract the RNA, put some adapters on that RNA so we can run it through a sequencing machine. And this machine will tell us the sequences of every organism in this plant, including cannabis. So next slide. And here are the results. So we took all that data, we looked at what was there, see if there's any known pathogens that could be matching up with the symptoms we're seeing and that are present in the infected plants and not present in the healthy plants. So if you look at this table, it turns out that the biggest correlation was to hop latent viroid. Okay, so we look at these bottom ones. These are all the infected ones, U1 through 6. Every single one of those had it. If you notice, the last one didn't. So this U6 was that middle picture we just looked at with those bright yellow symptoms. So those symptoms are not consistent with PCIA. Those are either genetic or something else. So next slide. You guys have likely never heard of a viroid. What is a viroid? Viroid is just a short strand of circular RNA. It's just RNA, there's no proteins or anything else. So for size comparison, if you look at this picture, the cell on the left, that's one of our cells, a eukaryotic cell. And we get smaller than bacteria, smaller than viruses, and then you have viroids. Very, very tiny. Next slide. And here's the genome. Very small genome, 256 nucleotides. You look at this picture, you're like, why is it called hop latent viroid? And so when viruses and viroids and pathogens are identified, it's identifying the crop it was first found in this case, hops. So hop latent viroid is considered a minor pathogen in hops. It does not cause severe symptoms as we see in cannabis. It does provide a small yield loss and some of the acid contents reduced, but not nearly as big of a problem as we see in cannabis. Next slide. Oh, can we do that? Thank you. And so this is just the phylogenetic tree for viroids. It's in the family Phosphopyridae, and it's a cocaine viroid. Next slide. So known host range, since it's not a major problem in hops, a lot of studies around the host ranges haven't been done yet. But what we do know, it is hops is a host, and possibly stinging nettle could also be a host of this disease. So if you're doing an outdoor grow situation, that's something you might want to focus on getting out of your space. Next slide, please. So now we have a correlation between hop latent viroid and the disease symptoms we're seeing in PCIA. So how do we turn that correlation into causation? The first step is we need to develop a genetic test so we can tell which plants are healthy and which plants are infected. So same sort of start, we take a cannabis leaf, grind it up, get the RNA out, do a test called RT-PCR, and that test just tells us, yes, this plant is infected, or no, this plant is healthy. Next slide. So once we have healthy plants and clean plants, then we need to do the last step. And for scientists, the way you figure out causation is something called Cox postulates. And all this is a set of experiments to make sure that what you think is causing the disease is actually causing the disease. So what you do is you find an infected plant, you make a pure culture of that infectious pathogen, and once you know that's all you have, that single pathogen, you infect that and inoculate that into a healthy plant, and then that healthy plant should become infected and show symptoms consistent with the PCIA disease. And for viroids, it's a little different. You can't grow them in pure culture like fungi or bacteria, so we made something called an infectious clone. And when this is inoculated into plants, it'll make the viroid. It'll make the disease that we see. So we did this. We tracked 2 different clones, pathogen clones made, and we also tracked buffer control healthy plants. And we waited 3 months to see what kind of symptoms we'd see. And next slide. And here are results. They're pretty stark. You can see on the left, nice healthy plants, much larger. The infected plants are very stunted. And when we took this picture, if we looked at the leaves, they were just starting to show signs of the malformation that's very consistent with PCIA symptoms, the yellowing of the leaves. So now we've gone that next step. So we've gone from correlation to causation. This experiment showed that HPDLD, or Hop Latent Viroid, is the cause of the PCIA symptoms we're showing. Next slide. And so now that we know what the problem is, we know for sure it's Hop Latent Viroid, how do we get this thing out of the cannabis plants and help the industry to increase those yields by removing it from the system? And Will's going to take over here. He's going to walk you through our system on how we cure our plants.
Okay, thank you, Jeremy. Hello, ladies and gentlemen. My name is Will Roberts. I'm an agronomist and researcher here at Heart Nursery, and I do all things research and development. And I'm going to tell you how we use plant tissue culture technology to eradicate pathogens in cannabis, specifically Hop Latent Viroid. So our patent pending process begins with a pre-treatment using small plants at our nursery facility. There are several different types of pre-treatments. Most involve extreme heat, cold, a few other kinds of things. But really the goal during this pre-treatment is to reduce the viroid load as much as possible before the next step of the process. But crucially, this pre-treatment does not in of itself cure the plant of any pathogens. So next slide. After the treatment, we bring the treated plants to our tissue culture lab. So I've been tissue culturing cannabis since we at DHN started our tissue culture lab back in 2014. We've really built up quite a bit of knowledge and expertise in plant tissue culture. Typically, nurseries like ours will use tissue culture technology as a means of propagating, so propagating more plants. So that's a very conventional, fairly straightforward process. But the universe of plant tissue culture technology includes all sorts of advanced techniques and technologies, a lot of it geared towards eradicating pathogens like Hop Latent Viroid. So about 1.5 years ago, we began doing virus eradication research to purify our genetics and hopefully produce certified clean plants. So we take the pre-treated plants from the first slide that I just showed you, and we cut off the very tip of the new buds, and this is called the meristem. It's a tiny, very delicate piece of plant tissue. It's usually about less than 1 millimeter across, sometimes much smaller. And you really need a microscope to see it, so that's what you're looking at here is how we use a microscope to dissect plants and just excise the very small meristematic tip. The idea here is this is the newest, youngest part of the plant and the least likely to be infected, the most chance of obtaining a clean plant. So next slide. These meristems are then cultured in petri dishes, test tubes, and larger vessels until they are large enough to test for Hop Latent Viroid, and then eventually we're going to bring those to the nursery. This process of culturing the meristems takes several months because remember, these are microscopic pieces of plant tissue that we're trying to regenerate into a whole plant. For instance, this picture here on the left, this plant is probably about 3 to 4 months old before it goes from that microscopic meristem into a decent sized looking plant that looks, somewhat like a normal plant. Next slide, please. Once we have plants that test negative, we began the process of rooting the clean plants and then bringing them to their nursery. These then become the foundation of our mother plants that we can then use to provide the cleanest, highest quality clones possible and really just be as sure as possible about how clean the material we're passing on to our customer is. And that's really been made possible by the research we've done around the viroid testing. Next slide. The inspiration for all this is the kinds of institutions available from many other economically important crops. Institutions such as Foundation Plant Services, which is up in Davis, and other facilities in the National Clean Plant Network. They work to identify, test for, and eventually eradicate different pathogens from crops as a service to these industries. So any economically important crop that suffers from systemic pathogen issues typically is involved in this sort of institutional testing and eradication. Next slide, please. And that's really what we at DHN are hoping to accomplish here. So cannabis, of course, does not have the kind of government or university support that something like grapes or berries or other crops have had. And I really think our announcement here this week is the start of a whole new era for the industry that will help us keep economically devastating diseases from spreading throughout cannabis and really help us get a hold on this before it really rises to truly bad levels. Next slide, please. And with that, I'm going to turn it back over to Dan.
All right, thanks, Will. So as a result of this work, we're announcing to the world that we're now offering diagnostic and consulting services to the industry related to HPLVD. We're also offering sanitation services to cure the disease. I think this is really important for everyone to understand. Until now, diagnostic testing for this have really not been available, and we're already seeing folks respond to this announcement by saying that that of course they don't have this, and their plants are disease free, or that their tissue culture practices are eliminating this pathogen. It's really important, I think, for farmers and industry professionals to understand that unless institutions are testing for this pathogen, there's really no good way for them to confirm that they don't have this, that they're not impacted by this. The latent part of hop latent viroid means that many plants can carry this disease asymptomatically, sometimes for quite a long time. It's something we've seen quite a bit out there in the ecosystem. So the only way to really know that it's not out there is to test for it regularly, sample and test for it regularly, and use good culture practices. Yeah, we're also extremely interested in following up this work with new investigations into new pathogens. We expect that this is just one pathogen. We expect that there are more out there. So if farmers expect that they might have something that's not this viroid, we would love to help you identify what it might be. So to that end, please reach out to Melanie Nash, who's included on the slide here, and she can help set you up with our team to get some services and help figure it out. All right, and with that, I'd like to turn it over to Q&A.
Yeah, so Dan, we have 4 questions in the queue. A quick reminder to all of the attendees, if you have a question, just message me directly using the chat function at the bottom. So in order, since you've been running this test, what is the percentage of plants that you've identified containing this pathogen?
Let's see, maybe Jeremy, maybe you could speak to that a little bit. I don't know if we have a tight number, but you could probably give a sense of how widespread it's been.
Yeah, and in the tests we've done on plants in the industry, we've noticed there's been about a 30% incidence of PCI in most of the samples that we've been doing. And I would like to point out, as Dan did, that a lot of these plants aren't showing outward symptoms of the disease. And so it's really important to get those plants tested and out, because you don't want a typhoid Mary sitting in your grow house spreading the disease to everybody else.
Great. Next question. Have you seen this pathogen more prevalent in specific gene pools, for example, OG cookie?
We have noticed that there seems to be a little bit of a strain differences. We haven't done enough experiments to say for sure which strains are affected more than others, but we definitely think there's a strain component where some strains are definitely showing more strong symptoms than others.
Okay, great. And number 3, would using fermented plant extract of stinging nettle increase the risk of contaminating plants that are sprayed with that extraction?
That's an interesting question. So a lot of times when they prepare these things, they're subjected to a lot of things that could likely inactivate the viroid, but I would have to see what extract you're using and how it's processed. And it's possible that whoever's growing that might not have this as an issue in their nettle. We could do some testing around that as well, if you're concerned.
And do we know how the viroid spreads from plant to plant?
Yeah, so viroids are pretty much every viroid is the major means of transmission between plants is mechanically. So any sort of nursery pruning, nursery cuts, even brushing up against an infected plant in the greenhouse and then going to a healthy plant and brushing against that. These things are very, very easily transmitted by any sort of abrasions or cuts. There's also some research that suggests that it could be transmitted through some insects. A little more work needs to be done there. And I just saw the question popped up. I'm going to answer now. And so I showed you the family tree. So this genus of viroids has been shown to be seed transmissible. However, we have not completed the test yet to confirm that in cannabis, but judging by some of the results we're getting out in the industry, it seems like it's most likely transmitted by seed. But we're following up with those experiments as well.
And
yeah, I think
go ahead.
Just to jump on that one a little bit, really any type of, so I think farmers should be especially careful. Obviously in the nursery, we're very diligent about and we've instituted new protocols to manage cross-contamination between plants. But I think in flower production, growers should be especially careful around pruning and training activities. That's going to be a highly likely source of contamination for those growers. And farmers that are doing dense plantings, I think, should also be especially careful. We've seen, anecdotally, we've seen some of the worst symptoms reported from very dense canopy growers.
Okay, 2 more questions. Does this viroid manifest the same way as hops as it does in cannabis?
Yeah, so it affects the same sorts of properties. So it decreases yields in the hops plant. It reduces a lot of the important, flavor chemicals that they are interested in brewing. We see the reduced yields. We see the reduced production of oils and important the cannabinoids. Like the plants aren't as sticky. The yields are reduced in fully grown out plants. So the symptoms are very similar. They're just much worse, much more affected in cannabis plants.
And in terms of impact, have we seen any difference in indoor versus outdoor crops or hydro versus organic?
I'll take that one. Again, I think this is anecdotal at this point. We haven't had enough industry-wide research to understand how exactly and to quantify how it's impacting growers. But anecdotally, it does seem to have, the symptoms do seem to be most impactful on dense indoor plantings. For whatever reason, we hear, there don't seem to be as severe symptomatic reports from like outdoor growers.
Okay, and a follow-up to the viroid transmission question. So is tool sterilization, alcohol dip, hydrogen peroxide, bleach, etc., does that decrease the rate of viroid transmission?
Yeah, so these sorts of sanitizers are what's used in viroids throughout all of ag, and it's important to use ones that are active on viroids. We're still doing research to figure out which the best ones are. Generally speaking, the more harsh, the better for the viroids. I think we're going to do some research and we can share that with everybody when we find some good findings. I think Dan and I have been working on that.
And have you found any human health impacts resulting from this viroid, either in hops or cannabis?
So this viroid does not affect people. It only affects plants. So there shouldn't be any detrimental effects toward humans because plants are infected with this disease.
Okay. And have you noticed changes in specific chemical profiles, such as drops in specific terpenes versus terpene alcohols or ketones?
Yeah, we haven't done enough pathogen studies to look at the yields and the specific losses in regards to each chemical, other than looking in the greenhouse and noticing that there's obviously less production because there's hardly any resins on the outside of the plants. But that's another thing we want to get to in our research.
Okay. And did the viroid mutate at the genetic level?
So the viroid we isolated from the plant and sequenced is the exact same sequence as the hop variety. Exactly the same nucleotide to nucleotide. So no mutation.
Okay, great. And that, if anybody else has questions, we have come to the end of the line for ones submitted so far.
I have a comment. I just want to say you guys did a great job, and this is amazing.
Thank you, Rob.
Yeah, thanks.
Yeah, thanks a lot.
And,
We're really excited
a
about
long
it.
time coming.
As a plant pathologist, I'm always excited about finding causes of diseases and trying to help growers out.
As a whole plant physiologist, I'm glad that you're excited about finding about diseases. So that's awesome.
Great.
Appreciate your knowledge base. It's outside of my realm of expertise, and I appreciate it.
Thank you. Thank you.
Okay, so Dan, any last comments before we wrap up for the day?
No, thanks again, everyone, for coming out. If there are any media requests, we'd be happy to do interviews or answer any follow-up questions that might come up. Laura's a good contact for that. And Melanie's email is on the list here, melanie@darkheartnursery.com for any service requests. And yeah, we look forward to keeping the good work moving forward. Thank you guys for your support, and we'll stay in touch.
All right. Thanks, everyone. Have a great rest of your week.